When we talk about gene cloning, we're essentially inserting a specific gene into a vector, like a plasmid, which can then be introduced into a host cell to express the gene. So this process is fundamental in many areas of biotechnology and research. In this case, we wanted to clone the human gene in BAS1 into Picha pastoris. However, for the gene to be expressed correctly, it needs to be inserted in the right orientation and within the correct reading frame. Then we need to make sure of two important things. First, that the gene is in the correct orientation, and then second, that it's in the right reading frame. If either of these conditions isn't met, the protein we want to produce might not form correctly. So there are many techniques that we can use to make sure about these requirements. So first, the in silico analysis. So in silico analysis will help us to know that when we are using a technique for assembly, we're making sure that the orientation of our gene and reading frame is correct before doing it on a lab. So this will increase the yield of the assembly process. If we want to do this analysis on the lab, we can use a restriction enzyme digestion technique. So we cut the vector and the insert with the specific enzymes and then analyze the fragments on an average yield. If the insert is in the correct orientation, the fragments generated by digestion with certain restriction enzymes will be different than if the insert is in the reverse orientation. So we have to choose enzymes that have asymmetric cut size relative to the insert and the vector chosen. With the help of a restriction map of a vector and insert, we can predict the expected fragment size if the insert is in the correct or reverse orientation. Then we can compare this expected size with the fragments obtained on the yield. For checking the reading frame, we can use enzymes that cut within the insert and the vector in such a way that we can obtain fragments indicating whether the reading frame is intact. Then compare the patterns of fragments obtained after digestion with the expected fragments based on a correct reading frame. If the insert is out of the frame, the obtained fragments will not match the expected ones. Other technique could be orientation-specific PCR. We design parameters that we only amplify a product if the insert is in the correct orientation. If we get a product of the expected size, the insert is in the correct orientation. Then it could be, for example, protein expression testing. So finally, we can perform a protein expression test, as we suggested in the project, to use ELISA to detect the protein. If the protein is in the expected size and function, the gene is in the correct reading frame and orientation.